Now we know about kingdom Monera. Monera is the kingdom in which all the organisms are prokaryotic types. Scientist Carl Woese first described the three-domain hypothesis. According to this hypothesis, Carl Woese discussed that there are three domains: one, archaea; one, archaebacteria; two, eubacteria; and three, eukarya. According to the hypothesis of Carl Woese, the Monera kingdoms can be divided into two separate parts, that is archaea or archaebacteria and eubacteria. Archaebacteria and eubacteria. Bacteria means the prokaryotic unicellular organisms. Now, bacteria under classes of the plant groups, but it is the prokaryotic type of organisms, that is the division of kingdom Monera. Monera is divided into two separate groups, that is archaebacteria and eubacteria. Archae means most primitive. Archaebacteria, what is the difference in between archaebacteria and eubacteria? Eubacteria is the bacteria type of organisms which are modern, but archaebacteria are not modern bacteria type. It is most primitive type of bacteria. Now we learn about the archaebacteria. Archaebacteria, what is the cell wall type? What is cell wall? Here the cell wall is made by only protein plus acellular polysaccharide. Protein plus acellular polysaccharide. Any type of peptidoglycan does not found in this cell wall, but in eubacterian cells, the cell walls are mostly covered with the chemical form, that is peptidoglycan, and the 16S rRNA nucleotide found in this archaebacteria. There are two types, either obligate or facultative. Their respiratory mode in the Monera kingdoms, they are anaerobic, and at the archaebacteria type, that is obligate anaerobic, and that is facultative anaerobic type of archaebacteria. They are divided into three types. One, methanogens; two, halophiles; thermoacidophils. Methanogens, halophils are thermoacidophils. All methanogens are autotrophs, but halophils are chemoautotrophs. And thermoacidophils live in the environment where the temperature is so high and the medium of this habitat is always the acidic medium, and most probably pH value at 2, that is thermoacidophils. Examples: methanogens, Methanococcus, Holococcus, and thermoacidophils is thermococcus. It is the features of archaebacteria, the division of kingdom Monera. What is eubacteria? Eubacteria is the modern type of bacterial cells which is under the group of Monera. That's I discuss here. That is bacterial cell. That is cell wall, cell membrane, that is plasmid, nucleate, then mesosome, that is lamellae, and this ribosome. Yeah, I discuss here about this type of cells present at the eubacteria group of Monera organisms. That is cell wall. What is the making component of the cell wall of eubacteria? The cell wall of these cells, that's prokaryotes, also made by the mucopeptide, peptidoglycan, N-acetyl muramic acid বা glucosamine, and N-acetyl muramic acid. I further say that mucopeptide cell wall, mucopeptide, peptidoglycan, N-acetyl muramic acid, N-acetyl glucosamine. According to these features, this component of making of cell walls, the peptidoglycan is the most important things which presents in the cell wall of eubacteria and mucopeptide. Why it is important? Where the mucopeptide is 90% at the present of cell wall, it is called gram positive bacteria, and mucopeptide is 10%, it is called gram negative bacteria. What is gram positive and gram negative bacteria? According to the theories and the experiment of scientist Christian Graham. The scientist make a pigment which is made by the Christian violet plus iodine. It is called Graham pigment. Those type of bacterial cells can pigmented by this pigment is called Gram positive, and those type of bacterial cells is not pigmented as this type of pigment is called Gram negative bacteria. Examples: Staphylococcus aureus Gram positive and Escherichia coli Gram negative. Here the another form I discuss here, that is the nucleoid. What is nucleoid? Nucleoid is the nucleic materials or promoting materials or DNA materials which are haphazardly spread out into the cytoplasm, and it is only made by the DNA, the bacterial DNA, which is separately spread out into the cytoplasm and action as the highly role at the reproduction is called the nucleoid. Further, it is said that the genophore, the bacterial nucleoid is called genophore. It is not bounded by the nucleus and nucleic membrane. Only DNA can not found in the bacterial cell. Another DNA is spiral-like found in some type of bacteria. It is called plasmid. Plasmids are three types: A plasmid, R plasmid, and Ti plasmid. A plasmid, R plasmid, and Ti plasmids. And plasmid plus DNA made the peculiar and the specific component of the bacterial cells is called episome. Next, mesosome. Mesosome is the most specific cell organ which is made from the cell membrane of the bacteria, eubacteria. It is act as the respiratory organelle of the bacterial cells. And various types of pigments and lamella present in these cells, and a ribosome is also present. It is type 70S type, which is made two separate subunits, that is 50S and 30S. So the structure of bacteria, eubacteria, I discuss here and represent here. Now, another form found in some of the bacteria, that is flagella. Where the flagella found in the bacteria, according to the patient of flagella, the bacteria are various types, that is here. Only one bacteria present is Monoticus. The bunch of flagella species is called Lophoticus. all surrounded flagellated bacteria, periticus. Both two pole of bacterium cells found in flagella is called amphiticus. And none of the flagella is present, is called the atticus. So we can say about the what is monoticus. Only single flagella is present in any side of the body of bacteria cells is called monoticus. Lopoticus, the bunch of flagella only inside of the bacteria cells is called lopoticus. Now I discuss about the shape and size of the bacteria. That is various types of shapes found in the bacterial cells. That is the rounded called coccus. That is rod shaped bacillus. Coma shaped, that is vibrio. Shaped like spiral, spiral. And the cuboidal, that is called Sarcini. Here the coccus, only one a single coccus found is called monococcus. Two numeral is called diplococcus, three numeral triplococcus, and the chain of coccus is streptococcus, and the bunches of grapes like coccus is called the staphylococcus. So the structure, shape and size of the bacteria we learn about this. Now I learn about the nutrition procedures of bacteria. There are two types of nutrition found in bacteria cells. That is heterobiotic and the autobiotic. That is heterotrophic and autotrophic. The nutrition of bacteria are two types: heterotrophic nutrition and autotrophic nutrition. Heterotrophic nutritions and autotrophic nutritions. In this type of nutritions, bacterial cells cannot depend the other type of cells or organisms. Those autotrophic nutritions of bacteria are divided into two, that is phototropics and chemotropics. Autotrophs and chemoautotrophs. When the food making procedures is depend only the abiotic component, that is light, it is called photoautotrophs, and the food making procedures only depend on the chemical substances, inorganic chemical substances, that is called chemoautotrophs. Heterotrophs also divided into two types, that is metatophs and paratophs. Metatophs, it is only the like as saprophytic depend, they make their food depending on the dead and decaying organic materials, and paratophs are the divided on the separate group of heterotrophic nutrition found in the bacterial cells. Now we learn about the reproduction of bacteria. There are three types of reproduction found in bacterial cells. One, vegetative. Two, asexual. And three, sexual. Vegetative reproductions are held by the various processes like budding, fission, segmentation, etc. Some of the bacterial cells can propagate their bodies by the budding, by the fission, or by the segmentation and produce the daughter cells, daughter bacteria also. Asexual reproductions are reproduction type which is called the spore making reproduction. Spores are various types, that is endospore and exospore. Many type of spore found in the asexual reproduction type of bacterium, that is conidia, gonidia, endospore, etc., mesospore, etc. Sexual reproductions are three types, that is transformation, transduction, and conjugation. What is transformation? Transformation means the bacterial DNA, dead bacterial DNA are transferred into the living bacterial DNA, or living bacterial DNA transferred into the dead bacterial DNA, and this DNA is replicated and produce the living bacteria. Charles Griffith first elaborate this type of reproduction. Visitors, that is are two type of, that is the rough, that is bacteria which is not covered by slime layers, and it inject the rat. Those type of bacteria is not covered by the slime layer, so it always killed by the antibody of this blood of rats, and the rat automatically live after injection of this type. Now smooth three type of bacteria which are covered with smooth walls, and when inject the rat, the rat is dead by their actions. But when it is living, it is living. But when it is dead, push into the rat. The rat is not fall on the harm, and it is also living there. They are also live in nature, and when the living two are bacteria cells and plus dead 3s mixed and push into the rat by the injection method, and this bacteria is killed by the bacterial action, because the living or bacterium DNA is transferred into the dead bacterial DNA of smooth 3 types is converted into the living R and is kill the rats. That is the transformation process. Transduction is the another process by which the dead virus, the bacteriophage virus, when adopt the DNA and transfer into the living cells of bacteria and produce the different daughter cells, it is called transduction. Conjugation, when two bacteria produced the conjugating tube and the nucleic materials transferred into another bacteria and produced the two separate bacteria, is called the conjugation. So all of the features I discuss here, that is the kingdom of Monera. And according to the thermal sensitivity, bacteria can be divided into 3 types. Thermal sensitivity, that is cyclophilic, bacteria, cyclophilic, mesophilic, and thermophilic. Cyclophilics, this type of bacteria can tolerate the huge low temperature, that is the cold habitat. This type of bacteria can found in the cold habitat. That's cyclophilic. And mesophilic, not the high temperature or not the low temperature, the mesophilics. And thermophilics, also the bacteria found in high temperatures also. That is, cyclophilics are 2 types, obligatory and facultative. And hence thermophilics also 2 types, obligatory and facultative. Those type of cyclophilic bacteria only live in the cold habitats, it is called cyclophilic, and some of the bacteria, cyclophilic bacteria, can live the another environment, not only the cold habitat, it is called facultative cyclophilics. And those of bacteria which are depend on the high temperature, is only high temperature, is called obligatory, but some of the thermophilic bacteria depend both the high temperature zones and the other zones, is called facultative. So I discuss here the